We're going to talk a little bit about the history of DNA and some of the important discoveries that led to our understanding of what it is and what it looks like. Now, you're not going to see a lot of history questions in general on biology exams, but this is one exception, and you are going to be tested on, and this is in the curriculum, on the contributions of various scientists to our understanding of DNA. And the reason is, it's a pretty interesting story, you know, how the work of several scientists over several years contributed to what we now know about DNA. And that's really how science works. It's how each scientist builds on the discoveries of previous scientists. Anyway, let's jump right in. The first guy you need to know about is called Phoebus Levene. There he is. Kind of looks like Kevin Kline, if you ask me. Anyway, his contribution was that he was the guy who discovered DNA and RNA, deoxyribose, or deoxyribonucleic acid, and ribonucleic acid. He had no idea why they were important or what they did in the cell, but he was the guy who first isolated these compounds. He also discovered that chromosomes were made up of DNA and protein. Okay? At this time, like I said, we did not know why DNA was important, but you know, we'll give him his due. He was the guy who discovered those two chemicals. The next guy we're going to talk about is Arthur Griffith. Now, he is important, or his experiment is important, because it proved that there is a chemical inside a cell that acts as the blueprint of a cell. Now, today we know that chemical is DNA, and that DNA acts as the blueprint of a cell. Back then he had no idea it was DNA, but he proved the existence of such a chemical that made a cell what it was. Now, he was working on finding a vaccine for pneumonia. He was a medical surgeon. It was during World War I, where he was losing lots of patients to pneumonia, and he was trying to figure out a vaccine for pneumonia. So it was kind of a side bonus of his discovery. He was working with two strains of bacteria, the R strain and the S strain. Now, the R strain was non-deadly, so I inject the R strain into the mouse survives. The S strain, on the other hand, was deadly. So if I inject the S strain into the mouse dies. Easy to remember because the S strain is deadly and that sucks. The S strain sucks, right? It's deadly. Okay, got it. Anyway, so he noticed that when you injected the S strain into the mouse died. When you injected the R strain into the mouse lived. So then what he did is he took that deadly S strain, but he heat killed it. So he heated it up really hot, enough to kill the bacterial cell. So then when it cooled down again, he injected into the mouse, and no surprise, the mouse lived. Because the bacteria was dead, right? So the next step he took was the interesting one. You can see that in this step he took heat-killed S bacteria and live R bacteria, injected them into the mouse, and the mouse died. So basically he was combining these two steps. And in each of these two steps the mouse lived. But why is it that when you combine the two, the mouse dies? Well, here's what he figured out. This is the deadly but dead S strain. So the dead bacteria that could kill the mouse, but it's dead, so it won't. However, inside that cell is a chemical that makes the cell what it is, and we know it's DNA. You can't kill a chemical, right? You can kill the bacteria cell, but DNA is not alive in itself. You can't kill it. So even though I killed this bacterial cell, I did not kill the DNA. And when I mixed these two together, the DNA was able to move from this dead bacteria into this live bacteria. And when it did that, it made this non-deadly bacteria deadly. So when we inject it into the mouse dies. So Griffith called this the transforming factor because it was able to transform the non-deadly bacteria into deadly bacteria. Okay? It acted like the blueprint of the cell. So he discovered the existence of such a chemical. Again, he didn't know it was DNA. Now we do. All right, the next experiment we're going to talk about was carried out by Avery, McCarty, and McLeod. Now, their discovery was important because it gave pretty good evidence that it was DNA that was the genetic material in the cell, or the transforming factor, and not protein. At the time, most scientists believed it was protein. So they pretty much repeated Griffith's experiment. So they took the non-deadly bacteria and the heat-killed deadly bacteria, mixed them together, and then they injected into the mouse. But before they did that, they added one step in two different trials. In one trial, they added protease before they injected into the mouse. Now, protease is an enzyme that breaks down the protein. Okay, so all of the proteins in that mixture in those bacterial cells would have been destroyed. So when they injected into the mouse died. Not surprising, knowing what we know, and that DNA is the transforming factor. You break down the protein, it has no effect on the DNA, does it? So the mouse would still die. In the second trial, before they took that mixture and injected into the mouse, they added DNAase. Now this is an enzyme that breaks down DNA. So, you break down the DNA, well, transformation is not going to happen then, is it? Okay, it's not going to, that DNA is going to be destroyed. It's not going to make that non-deadly bacteria deadly. So we inject it into the mouse lives. So these guys gave pretty good evidence that it was DNA and not protein that was the transforming factor. Okay, but still lots of people disputed that. Still people thought it was protein and not DNA. And the next experiment we're going to talk about is Hershey and Chase. And they put it to rest. They proved once and for all that the transforming factor, the blueprint of the cell, was DNA and not protein. Now they worked with bacteriophages. These are small viruses that infect bacterial cells. And here's what it looks like. And it's got a head and a body, and inside the head it's got the genetic material. And we know that it lands on a cell. And when it lands on a bacterial cell, it injects its genetic material into the cell. And it takes over the cell and uses it to make a whole bunch of baby viruses, and then it releases them. Well, that's kind of how viruses work. The question today was, is that genetic material protein or DNA? So that's what they were trying to prove. So here's what they did. They made 2 batches of viruses. I'm not going to get into the details of how they did this, but they grew 2 batches of viruses. One of them contained radioactive sulfur, which is only found in proteins. So they made viruses with radioactive protein. And in the other, they made radioactive viruses that had radioactive phosphorus, which is only found in DNA, so radioactive DNA. Now, I'm not going to get into the whole isotope thing or the chemistry of radioactivity, but elements that are radioactive are very easy to trace. So once they made these 2 batches of viruses, they let them infect bacterial cells. So over here, this cell infected the bacterial cell, it injected its genetic material into the cell, and when they tested for radioactivity, they did not see any. So what that showed is that protein did not make it from the virus into the cell, and therefore it's not protein that is acting as the transforming factor or the blueprint of the cell. On the other hand, when they did it with DNA, they let the viruses infect the bacterial cells, and then they tested for radioactivity inside the cell, and they found it. So that proved that DNA was passed on from this virus into this bacterial cell. And so therefore DNA was the transforming factor or the blueprint of the cell and not protein. Next guy we're going to talk about is Chargaff. Now Chargaff didn't really carry out one experiment, but he carried out several experiments, and he made some important discoveries about the composition of DNA. He was the guy that figured out that the amount of A in a DNA molecule is equal to the amount of T. Okay? And he also figured out that the amount of G is equal to the amount of C. Now A and G, these 2 kinds of bases are called purines. T and C, these 2 kinds of bases are called pyrimidines. He discovered that in any DNA molecule, the amount of purines is equal to the amount of pyrimidines. In other words, A plus G equals C plus T. Okay? Now it was at one time thought that all DNA molecules were consisting of the same amounts of A's, T's, G's, and C's, and we know that's not true. So he proved that that was wrong. He proved that, in other words, A plus T, we know that A and T always go together, or they're equal, equals G plus C. He proved that this is not the case. A plus T does not equal G plus C. The only way this could happen is if the DNA molecule was composed of equal amounts of A's, T's, G's, and C's, and we know that's not true. Okay, Watson and Crick. These guys are very famous in their day because these guys were the guys who actually figured out the structure of DNA, the double helix, the hydrogen base pairing between base pairs, the phosphate sugar backbone. They put it together, they took what was known about DNA and they figured out the structure. This is an X-ray diffraction, a picture of X-ray diffraction on a DNA molecule carried out by a scientist called Rosalind Franklin. So she was basically bouncing high energy X-rays off the DNA molecule and looking at the interference pattern, and this is the image that they got. So based on this, they figured that the DNA molecule was a double helix. And Watson and Crick, they knew from Chargaff's rules that the amount of A always equals the amount of T, and the amount of G always equals the amount of C, so they figured out that, hey, if A is equal to the amount of T, then A and T must go together. and G and C must go together. So they were the guys that put together the whole structure of DNA, and we know now is the actual structure.